Think about the split second you catch yourself before falling off a curb. Your body moves, but inside your skull, millions of cells are firing in perfect unison to catch that wobble. They do not wait for your command. Instead, they listen to each other with nanosecond precision. Yet many athletes spend years on boards and still shake when their focus slips. The disconnect isn't in the muscle. It is happening too slowly in the signal timing. This specific network acts like a librarian, sorting pages faster than anyone can read them all without losing track. Why do we keep training the body while ignoring the microscopic clock inside it that must tick in sync for true stability to emerge? But that connection requires something far more precise than just raw effort. Imagine an orchestra where every musician must hit their note exactly when the conductor blinks, or the harmony falls apart instantly. Inside your cerebellum, this is happening through spike timing-dependent plasticity. You have to synchronize high-fidelity parallel fiber inputs with Purkinje cells so they dance in lockstep. Think of it like a clockwork mechanism adjusting its gears in real time based on how much you sway without thinking. We aren't just moving limbs anymore. We are training the firing patterns of thousands of granule cells to dampen internal noise instantly. When those neurons talk in perfect rhythm, you get that sense of absolute stability. But what specifically triggers that exact synchronization window?
That synchronization window opens only when the brain predicts your next move before you even slip. Think of granule cell pathways as a filter sorting which signals matter. If they arrive too fast or too slow, the connection fades instead of growing stronger. This is spike timing-dependent plasticity, where physics becomes biology. We are seeing these cells fire in unison only when the vestibular loop matches the ground truth from your feet. Think of this process like tuning a radio while standing on a moving train. The signal stays clear only if you move with the broadcast, not against it. Direct brain observation shows this hidden synchrony, but there is a catch. That biological filter requires an external push to reach its maximum efficiency threshold during learning tasks.
So that external push isn't just force applied at any moment. It has to arrive exactly when your body is already swaying. Picture trying to catch a train that only opens its doors for a split second while moving. That is the window where parallel fibers fire relative to balance sway. When vestibular input hits mossy fibers during that precise interval, the synapse strengthens through spike timing-dependent plasticity. It is the brain editing static out of your sense of self without needing drugs. This happens in microzones where you map noise against stability. But here is the catch we need to solve now. Standard training fails because it misses that specific timing window entirely during movement. We are looking for a way to force that alignment during practice. So how do we force that alignment? It turns out the brain treats those milliseconds like absolute time zones. If a parallel fiber fires just nanoseconds too late, the Purkinje cell ignores it completely. We call this spike timing-dependent plasticity, but think of it like tuning a guitar string while the note rings out. It is too subtle for standard drills. We need to amplify the signal directly from the ground up using somatic input instead. Research shows that dynamic perturbations force these granule cells into sync much faster than quiet standing ever could. But there is a hidden catch in how we read those error signals without drugs. The climbing fibers hold a secret code written in calcium, but finding the right frequency to read it remains impossible. What happens when the brain refuses to tune that string? But what if that silence isn't empty space? Inside these tiny towers known as granule cells, a microscopic conversation is waiting to be decoded. Imagine trying to send a message across a crowded room where everyone is shouting at once. During balance training, these cells fire in rapid succession, but only if they arrive within milliseconds of each other. This is where timing matters more than strength. If you miss the window by a fraction of a second, the signal gets lost entirely. We see this happening in people trying to stand still on moving platforms where gravity keeps shifting. The vestibular system needs to talk to these granules faster than thought possible. But how do they amplify the weak signal when noise is high? That is where timing becomes everything. Imagine a crowded room with noise everywhere. The granule cells do not shout louder. They sync up perfectly, like a drum line marching in unison. This alignment triggers a signal that filters out background noise instantly.
It tells the Purkinje cell exactly what happened, cementing new pathways before you realize it. We see this occurs during training, but only if you stay within that specific window of focus. If you drift too far, the connection weakens before it can hold. They call this spike timing dependent plasticity. It happens in mere milliseconds. It explains why some days feel like magic, while others feel like stumbling through mud. Now we know how they amplify the signal, but here is the critical part nobody tells you about. What breaks when you try to speed up learning too fast?
What falls apart is the exact timing of those spikes. If you rush, the granule cells fire random noise instead of clean error signals. Think of trying to tap a drum while wearing mittens that slip every time you strike. The rhythm breaks instantly. We lose spike timing dependent plasticity because the neurons can't lock in to that precise frequency needed for correction. People practice constantly yet remain unstable because they are overdriving the connection before it solidifies. You cannot force the upload faster than the biological connection allows without breaking the synchrony. But there is a precise frequency window where that noise turns into signal again. We just don't know exactly where it sits for most practitioners. That leads us to look at something no one talks about.
That something happens when your brain learns to count the exact micro moments between steps. Imagine walking on a tightrope while listening to a radio station playing static in one ear and music in the other. Your cerebellum is the filter that separates the noise from what matters. It uses tiny spikes of electricity to mark time, like a high-speed camera freezing motion, so your body knows exactly where it lands without guessing. You don't need to force your muscles harder. Instead, you just need to find the pause inside your movement. Give yourself that split second between stepping onto something new and reacting. In that quiet space, the brain rewrites its own stability pathways while you move. That is how you turn wobbly focus into iron grip. The full research archive is at jmthomasofficial.com.